Welcome to the Research Highlights video series. These videos are intended to provide a summary of some of the research being conducted by members of the Hopkins Group at the University of Waterloo. This video highlights some of our recent progress towards using differential mobility spectrometry to assess ion microsolvation properties. For a more detailed discussion of how DMS can be used to determine ion solvent binding energies, please read our publication in the journal Analyst. Our mission in this study was to explore the use of DMS as a means of separating isomeric forms of quinoline-based drugs and to investigate the effects of ion-solvent interactions on DMS ion trajectories. 5 pairs of isomeric quinoline-based drug candidates were chosen for a combined experimental and computational study. Solutions of the quinoline derivatives were infused into the DMS cell containing nitrogen gas seeded with water vapor. As the quinoline molecules transited the DMS cell, an asymmetric AC voltage was applied to planar electrodes on either side of the ion channel. This results in a zigzagging off-axis migration of the quinoline trajectories, which can be corrected via application of a DC compensation voltage. By monitoring ion transmission as a function of the AC and DC voltages, we are able to determine whether the molecules interact strongly or weakly with the DMS atmosphere, or if the ion is essentially non-interacting. Since different isomers exhibit subtle differences in their physicochemical properties, the positional isomers of the quinoline derivatives can be separated based on their interactions with the DMS collision environment. Here we see that 8 substituted quinolines interact much more weakly with the water-seeded N2 environment than do the 6 or 7 substituted isomers. Interestingly, if we plot the compensation voltage minimum against the calculated water binding energies for each quinoline derivative, we observe a strong linear correlation. In other words, DMS measurements facilitate quantification of solvent binding interactions. This provides a microscopic view of incipient drug solvation. For more information about the Hopkins Group members and the research that we conduct, please visit the research group website, which is hosted by the University of Waterloo Department of Chemistry. Thank you for your time and your interest in our work. See you next time.